Hi, my name is Saraí Rivera and I'm going to present to you the meiosis simulation video. I'm going to begin with the first step, which is interphase. In this step, the chromosomes are replicated. 42 chromosomes are expected to be present. The clones of these chromosomes stick together and are called sister chromatids. After DNA replication, 92 chromosomes are expected to be present. What keeps them attached is called a centromere. The second step is prophase. During prophase, the nuclear membrane starts to break and spindle fibers begin to form. You see? These are the spindle fibers. The homologous chromosomes come together. These are called tetrads. There should be 8 chromatids in all. Crossing over occurs now. So chromatids begin to break into segments and they are exchanged. So here you can see the chromatids are being exchanged. In each tetrad, overlap chromosomes come to form, so one allele can be traded between 2 non-sister chromatids. Now we move into metaphase, where we're going to line up the tetrads so that the centromere points towards the opposite poles. During anaphase 1, which is what I'm about to show, only the tetrads separate. You see, the spindle fibers separate, but the tetrads, by pulling homologous chromosomes, keep pulling homologous. Ay, otra vez. Now I'm going to show you anaphase 1. During this one, the spindle fibers separate the tetrads by pulling the homologous chromosomes toward the opposite sides. but you see the sister chromatids, they remain attached to the centromeres. Now I'm going to show you telophase 1. During telophase 1, the chromosomes continue to move to the poles. I'm about to draw the poles right now. Now, as you can see, two daughter cells are formed when cytokinesis occurs. Now the question is, are the two daughter cells identical in genetic makeup? Well, they are, because they have decided, or at least divided, the genetic material evenly. Now, I'm going to show you the next step, which is prophase 2. During prophase 2, the chromosomes may condense and become visible, and the nuclear envelope dissolve. So let's say there was a nuclear envelope right here, and it starts dissolving till it will eventually disappear. This is the other pole. You see, these are the spindle fibers. Same thing here, the nuclear envelope starts to dissolve. And as you can see in each new cell, the centrals move to the poles and new spindle fibers are formed. Now, we're gonna move to metaphase 2, where the spindle fibers are attached to each centromere. And the sister chromatids line up at the center with the centromere between them. Well, now we're gonna continue to anaphase 2, where you see the centromeres that join two sister chromatids start to, give me 1 second, they start to break. And the sister chromatids move towards the opposite poles, like this. Because the crossing over that occurred during prophase 1, the sister chromatids are genetically different. You see? Right now, this is the last step, telophase 2. Now, as you can see, the spindle fibers have disappeared, and then the nuclei form at opposite poles of the cell and the nuclear membrane has just started to reappear. As you can see, cytokinesis has just happened to reoccur and has created a total of 4 daughter cells, aka 4 haploid daughter cells. Now you may ask, are the genotypes of any of the daughter cells identical? Well, they're different because they would have 1/2 of the genetic material as the parent cell. Now, the result is 4 daughter cells, and as you can see, each with a haploid cell of chromosomes. Thanks for watching.